Welcome back to G Fun Facts Online. Today, we are going to dismantle one of the most persistent metaphors in modern science. We often describe the human brain as a computer. It is an easy comparison to make. We see a network of wires in the form of axons and dendrites. We see switches in the form of synapses. We imagine information being processed like binary code, ones and zeros flashing in the dark. But this metaphor fails to capture a fundamental biological reality that separates us from our machines. In a silicon chip, the hardware and the software are distinct. The electricity that runs your laptop is just a utility. It powers the circuits, but it is not the information itself. The brain is different. In the brain, the hardware is intimately consumed by the software. The energy that powers the brain does not merely run the machine, it is the machine. At the heart of this bioenergetic unity lies a molecule that every high school biology student has memorized, but few truly understand: adenosine triphosphate, or ATP. For decades, ATP was typecast as a simple cellular battery. It was viewed as a passive currency, something burned to keep the lights on and the sodium-potassium pumps running. But in the last few decades, and culminating in breakthroughs as recent as 2025, a completely new picture has emerged. ATP is not just the fuel, it is the message. Today, we are exploring the complex dual life of ATP in the brain. We will journey from the microscopic machinery of mitochondria to the high-level circuits that control our breathing, our movement, and our pain, revealing how the brain uses its energy currency to talk to itself. Let's begin with Part 1, the bioenergetic foundation. To understand how ATP works as a signal, we first have to appreciate the sheer economic constraints of the organ inside your skull. The human brain represents roughly 2% of your total body weight. However, it claims 20% of the body's oxygen and glucose. This is a staggering disproportion. If your laptop consumed 20% of the electricity in your entire house, you would think something was wrong with the wiring. This immense metabolic demand is not evenly distributed, and this leads us to the concept of the brain's dark energy. When neuroscientists first started imaging the brain, they expected to see a massive spike in energy usage when a person performed a task, like doing math or looking at a picture. They were surprised to find that the energy consumption barely budged. It turns out that the vast majority of ATP is consumed not by these evoked spikes, but by the resting state. This is the housekeeping. This is membrane repolarization and protein turnover. It is the cost of keeping the machine on standby. Because generating action potentials, the actual electrical spikes of thought, is so metabolically expensive, neural circuits have evolved to be incredibly efficient. This leads to a theory known as sparse coding. The theory suggests that at any given moment, only a small fraction of your neurons are highly active. Energetic constraints force the brain to represent complex information with a minimal number of active units. It is effectively compressing data to save ATP. If too many neurons fire at once, the brain literally runs out of energy to sustain the ion gradients, and the system crashes. So how does the brain get this fuel? The classical view was simple. Neurons take up glucose directly from the blood. But biology is rarely that simple. We now look to the astrocyte neuron lactate shuttle, or ANLS. This hypothesis paints a picture of deep cooperation. Astrocytes are the star-shaped glial cells that actually outnumber neurons in many parts of the brain. For a long time, they were thought to be just glue, holding the neurons together. But we now know they act as metabolic hubs. Astrocytes extend long processes called end feet that wrap around blood vessels. They siphon off glucose from the blood. But they don't just hoard it. When neural activity rises, astrocytes convert this glucose into lactate via glycolysis. They then shuttle this lactate to the active neurons. The neurons take this lactate, convert it to pyruvate, and feed it to their mitochondria. This coupling ensures that energy production is spatially and temporally matched to synaptic activity. It is a just-in-time delivery system. Speaking of mitochondria, we need to change how we visualize them. We usually see them in diagrams as static beings floating in the cell. In neurons, mitochondria are mobile power plants. They are dynamic organelles that travel along cytoskeletal tracks, specifically microtubules. They physically move to dock at sites of high demand, the synapses. Synaptic transmission is an energy-intensive process. Packaging neurotransmitters, fusing vesicles, and recycling membranes requires a burst of local energy. If the mitochondria are not there, the transmission fails. Furthermore, mitochondria do more than provide power. They suck up excess calcium, helping to reset the synapse for the next signal. If mitochondria fail to dock at the synapse, neurotransmission falters. This is a defect we now see in the early stages of neurodegeneration. The logistics of energy are fascinating, but the story gets truly strange when we move to part 2, the machinery of purinergic signaling. In the 1970s, a researcher named Jeffrey Bernstock proposed a radical idea called purinergic signaling. He suggested that ATP could act as a neurotransmitter. He was met with intense skepticism. The scientific community asked, why would the body use its fuel as a signal? It seemed like spraying gasoline to signal a fire. But the answer lies in evolution. What better way to signal a cell's state than by releasing the very molecule that represents its vitality? If a cell is bursting with energy, or if a cell is damaged and leaking, ATP is the most honest signal available. We now know the brain is studded with sensors for ATP and its breakdown products. We categorize these into 3 main families. First, we have the P2X receptors. These are the fast lane. They are ionotropic receptors, which means they are channels that open immediately when ATP binds. They allow sodium and calcium to rush into the cell, mediating fast excitatory transmission, much like the classic glutamate receptors. Second, we have the P2Y receptors. These are the modulators. They are G-protein coupled receptors. When ATP binds to them, they trigger slow, cascading changes inside the cell, altering sensitivity, gene expression, or metabolic state. Then, we have the P1, or adenosine receptors. This is the brake. As ATP floats in the synapse, enzymes called ectonucleotidases strip its phosphates off one by one. ATP becomes ADP, then AMP, and finally, adenosine. Adenosine acts on P1 receptors to inhibit activity. It signals to the brain, we have burned too much fuel, it is time to rest. This is the mechanism behind sleep pressure. It is also why caffeine works. Caffeine is simply an adenosine blocker. It puts a piece of tape over the sensor so the brain cannot realize how tired it is. But how does ATP get out of the cell to hit these receptors? One way is co-transmission. ATP is often packed into synaptic vesicles alongside classic neurotransmitters like GABA or glutamate. When the neuron fires, it releases a cocktail of signals. But there is also glial release. Astrocytes communicate primarily via calcium waves and ATP release. They do not use vesicles in the traditional sense. Instead, they use connexin 43 hemichannels or pannexin channels. These are literal gates in the membrane that open to let ATP flow out into the extracellular space, modulating nearby neurons. This is chemical broadcasting. Now, let's move to part 3, ATP signaling in action. The true power of bioenergetics is revealed when we look at specific functional circuits. Let's start with something vital, breathing. Deep in the brainstem lies the pre-Botzinger complex. This is the pacemaker of respiration. It keeps you breathing without you thinking about it. Now, consider the problem of hypoxia, or low oxygen. If oxygen levels drop, neurons typically silence themselves to survive. They shut down to conserve energy. But if the respiratory center silences itself during low oxygen, the animal dies. It stops breathing exactly when it needs to breathe most. The brain solves this with ATP. During hypoxia, the brainstem releases massive amounts of ATP. This ATP binds to P2Y1 receptors on the rhythm-generating neurons. Instead of silencing them, it excites them. It forces an increase in the breathing rate, causing gasping. It is a fail-safe mechanism. The metabolic distress signal, which is the dumping of ATP, becomes the physiological rescue command. Interestingly, research shows species differences between mice and rats in how this circuit balances ATP excitation and adenosine inhibition, highlighting how evolution has fine-tuned this life-or-death system. Next, let's look at sensory modulation, specifically in our sense of smell and pain. In the olfactory bulb, ATP acts as a gain control knob. Olfactory sensory neurons release ATP to activate nearby glial cells. These glia then release their own ATP to modulate mitral cells, which are the output neurons. This feedback loop allows the nose to adjust its sensitivity. It prevents saturation from strong odors while boosting weak signals, ensuring we can smell a rose in a garden or a gas leak in a storm. In the spinal cord, ATP is a key player in nociception, or pain signaling. This is where things can go wrong. Following nerve injury, microglia in the spinal cord upregulate a specific receptor called P2X4. When stimulated by wandering ATP, these receptors trigger the release of brain-derived neurotrophic factor, or BDNF. This makes pain-sensing neurons hypersensitive. This is a major pathway in neuropathic pain. Essentially, it is a metabolic signaling loop gone wrong, treating the system as injured long after the initial wound has healed. Then there is motor control and the basal ganglia. This acts as the command center for voluntary movement. We know that dopamine regulates the go and stop pathways here, but ATP and adenosine are the shadow regulators. In the striatum, adenosine A2A receptors and dopamine D2 receptors are often found physically stuck together as heteromers. They are antagonistic. Adenosine inhibits the D2 signaling. This has massive implications for Parkinson's disease. In Parkinson's, dopamine is lost, but if we block the A2A receptors with drugs or even simple caffeine, we remove the brake on the remaining dopamine signaling. This helps restore movement. This makes purinergic receptors a prime target for the next generation of Parkinson's drugs. Finally, we arrive at Part 4, the new frontier. In a stunning discovery published in 2025, researchers found a third role for ATP, distinct from energy and signaling. At the high concentrations found inside neurons, which is in the millimolar range, ATP acts as a hydrotrope. A hydrotrope is an amphiphilic molecule that helps keep hydrophobic proteins soluble in water. It prevents them from clumping together. The study showed that high ATP levels prevent the aggregation of proteins like alpha-synuclein, associated with Parkinson's, and amyloid beta, associated with Alzheimer's. This implies that in neurodegenerative diseases, the problem is not just that neurons run out of gas. As ATP levels drop, due to mitochondrial failure, the cytoplasm literally becomes more viscous. It loses its ability to keep proteins dissolved. The toxic protein plaques precipitate out of the solution because the hydrotrophic shield has failed. This fundamentally changes how we view Alzheimer's. It suggests it is a solubility crisis caused by an energy crisis. In conclusion, the study of bioenergetics and ATP signaling closes the loop on the mind-body problem. It shows that our highest cognitive functions, our memories, our sensory experiences, our very will to move, are not abstract software running on a hardware substrate. They are biological processes deeply rooted in the metabolic fluxes of life. From the astrocyte feeding lactate to a firing neuron, to the desperate release of ATP by a hypoxic brainstem, to the hydrotrophic shield preventing Alzheimer's plaques, ATP is the protagonist of the brain story. As we look to the future of neurology and psychiatry, the targets will likely shift. we will move from correcting chemical imbalances of neurotransmitters to restoring the bioenergetic signaling that sustains the vibrant, electric life of the neural circuit. Thank you for listening to G Fun Facts Online.